Okay, everybody, welcome to this little workshop that we have before the meeting. How many people are here for the meeting, actually? So about half of us, and then the rest of us are some locals. So I'm Jason Williams. I'm here at Cold Spring Harbor. I'll be at the meeting with you folks tonight. There are probably a couple others that have registered. I see even now people walking in. So we'll let them gather together. And also, we are doing this online. So in fact, there are people that are watching me now on our various internet sites. So I'm going to give a little bit of an introduction to iPlant. The introduction I'm going to give is actually pretty short, even though I probably have way too many slides, because I'm actually going to give a more thorough introduction tonight at the meeting. So if you are new to iPlant and you need a lot more in depth, there's some online links. For those who are watching online, if you're watching on live stream, then you should be all set. If you are watching through Adobe Connect, so you're in the meeting room, if you look on the actual window where I'm displayed right now, you can choose the quality of your video, high, medium, or low. So depending on your internet connection, you can adjust that. If you've experienced any kind of technical challenges, you might want to move the quality down because it's HD, or you could always switch to the live stream. The wiki is circulated, so you should go to that wiki site. I'll put it up a little bit later. And if you have questions through Adobe Connect, you can go ahead and post those questions there. I'll also be checking the Twitter account. I guess I'll put that up. It's at iPlant Collab, where you can search Twitter, or you can go to the Ask forums. So we'll have some ways to ask questions. Also, the event will be recorded and you can watch it online. So also people who are here today, you can watch things online when it's done. So I'm joined by a couple of people. So Mike Schatz is going to be our first speaker in a moment. He's going to take us through the genome assembly, and then Josh Stein is going to take us through annotation. I'm going to go through RNA-seq towards the end. Also, we have some of our friends and collaborators here. So Marcella is going to take us through Grameen, a little bit of a demo a little bit later. I don't think I saw Sunita yet, but she's going to talk about K-base later on. So we have a number of things. We saved the best seats in the front for those who came a little bit later. But I'm going to try to keep this on time with a little bit of an introduction to what iPlant is about. So iPlant, the iPlant Collaborative actually got its start. The kickoff meeting was at Cold Spring Harbor, but it's been around for 5 years, and now we're beginning year 6. And we have a lot of resources that we hope we can offer anybody who's working in life sciences. So this is a plant meeting, and obviously we started with plants. But the tools are rather generic, so everybody can use them. But in order to be able to build what we have built over the past several years, you really have to have a vision. And so our vision is to enable scientists of all types to use cyber infrastructure to advance their research. And we'll talk about that, and obviously we'll hear about that and see that throughout the course of the workshop. That's a big job because, and you need vision for it, because it means planning for technologies and planning for things that you may not have anticipated. So my little cartoon there is of a place that's not far from here. Well, some of you might be from New York. I am over the Verrazano, the New Yorker cartoon. Someday this will all be infrastructure. And sort of looking at juxtaposed here with the first image of the double helix. And kind of the question is, in my mind, how could anybody thinking about DNA from the biochemical perspective 50 years ago ever think that today what it means to do biology is sitting in front of a laptop and being able to get information from your data, be able to manage data. So really, biology has changed a lot, and we need new tools in order to get our research done, and we need to come up to speed on how to do that. So you need to build a bridge there, and that bridge itself is infrastructure. And so that's why this whole project is a cyber infrastructure project. So what is cyber infrastructure? There are many different definitions, and it depends on who you talk to. So one of the persons who gives this talk is from the computing side, and he will have 30 slides about processors and things like that, that maybe as a biologist goes over our head. But really, cyber infrastructure is what you're doing right now. It's the computing systems, the software, the data management, all those underlying elements. Cyber infrastructure, like all infrastructure, isn't necessarily sexy. It's not something that you see, but it matters, right? That when you land in your airplane, there's actually an airport for you to land at. It makes a difference, right? But those are all the elements of cyber infrastructure. And also people ends up being one of the components, by definition, of cyber infrastructure. Knowing who it is that you can go to for help, and having a workshop like this is part of the people side of the whole enterprise. But building cyber infrastructure is not a project that an individual lab or organization can do. I mean, there are plenty of labs that put out great software and as individual pieces, but building something that everybody can use in their own way and building a platform means that you actually have to have an entire community kind of coalescing around what are the objectives, what do we need to build, how do we prepare to build things that 10 years from now will still be useful. That's a little bit of a bigger job. So in terms of where we were, where we've come from, as I said, 2008 was initial funding. And the first year and a half, almost 2 years, was really scoping and learning from the community, what did you need to build? I'll talk about more of those things in depth tonight at the actual meeting. But over there, and these will be updated, so I'll show the latest numbers. But we've been able to allow lots of people access to a cyber infrastructure, to computing systems, to data, and let them use it the way that they need for their science. And really, our overall goal is to open up some of the bottlenecks that you have to doing your research. So we've been able to do that to a large extent, and you'll see that and hear more about that a little bit later on. Importantly, we were just renewed by NSF. So that means that if you're new to iPlant or if you've been with us for a little while, you still have access to all of these resources that we built, plus what's coming down the pipe. So this is an important message, I think, in the sense that it doesn't really make sense, and I've seen this story a dozen times, for individual labs or individual projects necessarily to always buy new infrastructure and buy new computers and buy new hard drives. Of course, that's always going to be appropriate to a certain extent. But if you can have some shared resources that you just pull into when you need them, that makes a lot more sense. So that's part of, I think, the message that NSF, or the goal NSF had when they originally called for proposals. And so we've been able to accomplish that. And some of our new features, or our new going forward, will be still being responsive to the community. So we have a scientific advisory board that helps us understand what the needs of all the biologists are, and then we can better serve them. And of course, our scope has broadened too, because this is something that's useful to all life sciences. So there are a number of different things. I'm going to try to wind up, even though there's some more slides here, and they'll all be online. But essentially, what do we provide for you? And my one key message to anybody that's wondering what iPlant is, and as you're going to see through some of the demos what you can do, is it's a platform, and really it's there for you to take what you need from it, and then you don't have to really worry about using the rest. We're not trying to lock people into using iPlant for every single thing, but we do want to help you identify what are the pieces of iPlant that lock into what the needs of your individual scientific goals are, or your lab's goals are, your project's goals are. So whether you need data storage and access to computation, whether you have a new set of tools that you want to move into the iPlant environment, whether you need to have hosting or use any of these other resources through our APIs, through cloud computing. So these are all things that are accessible to you, and we're going to touch on some of them today. That's another caveat of this workshop. Our normal workshop is a 2-day workshop. Many people end the workshop saying, I wish it was a week, because it's a lot of stuff to cover. So there's no way we're going to make people experts in anything in just a few hours. We haven't got that type of technology, but we can get you started. And so these are things you just need to be aware of. And as I'll show you again, these are all on the wiki, so if you miss any slides or want to go back, it's there. We're going to be working, and I'll take you really quickly into the discovery environment, which is the biggest and the broadest sort of platform. It was built really with the everyday bench biologist in mind. As we know, I didn't do the poll yet, but I do know that from surveying the people that are attending online, most of us did not go to school to be computer scientists, and then fewer are bioinformaticians. So a lot of tools, a lot of things are command line, a lot of things are just already, you need to learn Linux and R and all these other things before you can even touch them. The discovery environment is a platform that allows you to do a lot of those things in a much simpler and intuitive interface. So we're going to get started with that. And the other thing that we're going to be using throughout the day is Atmosphere, especially what Josh is doing. So that is a cloud computing solution. How many people have used cloud computing? Just by a show of hands. Okay, and you're like ashamed of it because you're not even raising your hands really high. But probably, so if I ask the question, how many people use Netflix or Facebook or Amazon or anything like that, everybody's hand should go up because they're all forms of cloud computing, and this is just a biological cloud computing infrastructure. We'll see what that means. You have all sorts of acronyms and words and buzz terms, but essentially it's another way to access in a customized environment and do your science. The other thing that you have is your data store. So being able to store your data already, that's like the lowest level of what most people want right away. You've got your sequences back from whoever sequenced your data set, and they tell you you've got 3 weeks to download it, and then it's going to disappear. And already you're like, well, how do I get it? Are they mailing me a hard drive? Do I have to sort of download it somehow? But when you get your iPlant account, you already have 100 gigabytes of space, and that's, if you need a terabyte, you can go ahead and ask for a terabyte. If you need more than that, just a little bit of justification. But right now, it's all sort of free. There's no cost to it. So I would go ahead and feel comfortable putting your data there. And then through the iPlant data store, as you'll see, and we'll use it many different ways, it's fast, and it's also all interconnected, so you can really get a lot of things done quickly. There are a lot of other things coming. If I add more slides to my talk tonight, I'll get to more of them. But we are just expanding what we're able to offer you by interoperability with other data sets, other peer resources out there to make the best experience for our users. And so it's really critical to all of us that you get what you need out of it and that we listen to what you want. So with that said, I think that's the end. There's a little bit of acknowledgement of the large group of people that are behind what iPlant is, what iPlant does. I'm going to take you into the first exercise, which is with the discovery environment. I just want to get you oriented, take you a 5-minute tour of what the discovery environment is, and then Mike is actually going to show you how you can do one of the first things that a lot of people are interested in now is looking in genome assembly. I think the wonderful thing about what's happening in biology now is that many things that were really, really complicated to do and that you needed a whole consortium to do are now becoming almost single-person activities. A single technician in front of an Illumina machine, a single bioinformatician. So there's really a lot of things that we have the power as individual bench biologists to do. And so giving you access to that is what we want to enable. So let me take you into the discovery environment, show you how to get access to things right now and orient you. So let me actually open up my computer here and tell you a few things. All right, so first, what I want to emphasize is that we have this wiki. So if you have your handout, and I should just grab a handout or somebody will hand me one. I'm going to borrow this for just 1 second just so everybody knows. These are the handouts, and then some of them, you have one of the nice ones that are stapled nicely. Some of them have them that are stapled the wrong way. This is the handout. If you are online, this is posted on the Adobe Connect room. And if you are watching a live stream, this will be on the wiki, so we can get the links to you. But this CSHL PBG wiki, so I'll put that up somewhere if you can't see that, but it's iplantc.org/CSHL PGB wiki, all one word, kind of there for you. You just want to get to the wiki so that you see where all the slides will be posted. We'll put that up several times, so don't worry if you don't get it right now. Just want to know that it's there for you. Thanks for that. So the first thing we need to do is actually get into the iPlant discovery environment. Everybody should have an iPlant account. If you don't have one, you can go ahead and register. So let me take you through getting inside. So first, we'll go to the iPlant homepage. You can either Google it, or it's iplantcollaborative.org. So this is the iPlant homepage. And from the iPlant homepage, if you're pretty familiar with this, you can already click on Discovery Environment to log in on the button there. If you're kind of new, you might want to go to this sort of login and register, which is at the top there. Let me zoom in so you can see. And this is the user portal. So you can also log in here from the user portal. If you don't have an account, you can go here, user.iplantcollaborative.org, and you can get an account. If you don't, it takes a few moments. Support is pretty responsive. And I'm going to go ahead and just click login. Okay. When you log in, you have all your services. And I'm going to go ahead and click on the discovery environment. And when I click on it, since I'm already logged in, and it's probably going to open to the last things I was working on, so I'm going to close that for just a moment. And I'm just going to give people a moment to get into the discovery environment. Now, I'm here, but also Christos is in the back here. Chris is also with iPlant. And also Josh and Mike, at least for this moment. When one of us is not talking, you can always just raise your hand if you need something, and we'll zoom around to help you. But I just want you to get here. So let me orient you, and then we'll have Mike come on. So in the discovery environment here, there are a couple of different things. There are some settings and some help up here on the right-hand side. You don't need to worry about that so much, although through here there is a little bit of an introduction. So if you're bored for a second, or if you forget something, you can go to the introduction. I'm just going to take you through it myself. If you click on the data window, the data window gives you access to everything that is in your iPlant data store, so all the files that you've saved. Most things here operate like a drag and drop environment. So your home folder, which is your iPlant username, is here, and that would contain all of the files and folders that you need. Most things work, as I said, with drag and drop, where you can move one thing from one place to another. If you open up an individual folder, the files are inside, and I've opened up a big folder. Let me open up something from my home folder. You have the ability to rename files, to move files around. So, I mean, it's pretty intuitive. You can click on something, click on an individual file, and you'll get a preview of what that file's contents are. So all of those things are there for you. So one thing I want to point out is, very often, I know I certainly will, on this left-hand side, there's a community data folder, and many of the files that we're going to be doing, certainly with my demo, are staged. So many things you'll be coming into community data. I'm going to close the data window for a moment. Back here, there are apps. So in apps are actually all the applications that are in the discovery environment. So what you want to think of the discovery environment as a tool or a workbench, and it has various bioinformatics apps in there. To make a long story short, almost any bioinformatic tool that you use in the command line, almost any one of them can be integrated into the system. So right now there's a catalog of about 430. If there's something missing, you can add that in, and we can talk about that later. But essentially, if you want to use an app, let's say Muscle, I've favorited here, which is an aligner. You start out with your app, and you sort of give your job a name, your analysis a name, and then it has this sort of accordion menu of various options, and you go through and you can select the options. You really see that when Mike is using that. And the final thing that you need to know to get started is here under Analyses. This is a history of all of the jobs that you've done in the discovery environment. And this is useful for a number of reasons. When we're in the lab, if you didn't have a lab notebook, you probably wouldn't last very long. You'd be asked to leave. But when we're on the computer and doing bioinformatics, our first instinct, mine certainly, is to press as many buttons until I feel that things work. And then I go back and like, well, what did I do to get that graph or to get that to come out? So this is a history there. And if you click on the app, you can actually go to the results of that analysis. You can relaunch, you can cancel. So it's just a history that's there. So I'm going to close this, and I'm going to log out. And we're going to turn it over to Mike. And he has some slides to go through. and a little bit of a demo. And our first subject is this genome assembly. So let me just log out.